One moment. Cool. Okay. Just some general information. This is the 15-week MCAT course. I believe we're on week 13 now. There's the GroupMe link, the Instagram handle. There's our YouTube page. It has all of our lectures as well as sectional videos from each of our lectures. We do 3 lectures a week, Thursday, Friday, Saturday at 8:30 Eastern time. And we also have office hours every Sunday at 8:30 Eastern time as well. Okay, so we're going to get started with a quick introduction to immunity. This is probably going to be a pretty quick lecture relatively. There's not too much content on this, so I will try to elaborate as much as possible, but I didn't want to put too much on here that wasn't that didn't pertain to the MCAT. So I just tried to fit on the slides what is expected of you when you take the MCAT, specifically for the biology section. So let's get started. What is immunity? The immune system is the bodily system composed of organs, tissue, and other substances that protect the body from infection and disease. So the immune system is comprised of 2 systems, the innate and the adaptive immunity. Adaptive immunity is also sometimes further divided. It really depends on what study material you use. Sometimes you'll see adaptive immunity divided into humoral immunity and cell-mediated. Don't get too stressed out over which division is used in which specific resource. Just as long as you understand specifically the components of each, like you understand what the cells are doing, you understand specifically what each job is, you don't really have to worry too much about, oh, is it adaptive? Is it humoral? Is it cell-mediated? So usually when I teach it or when I learned it, I just learned that it was split into innate and adaptive. So that's probably how I'm going to frame the lecture, just within those 2 subsets of the immune system. And both of the immune systems work together to provide a robust response against foreign invaders. So it's usually a combination of both. It usually starts with the innate immune system, and then the adaptive immune system takes over. And we're going to get into that in a little bit. I put this graphic over here. It's just a basic sketch of the immune system and part of the lymphatic system. Okay, so innate immunity. Innate immunity is comprised of multiple organs, proteins, and cells. The innate immune system mounts a quick, non-specific response to a wide variety of pathogens. So that is the important part about the innate immune system. It is pretty much the first line of defense for the body. So it mounts a quick response as soon as it recognizes that there's a foreign invader in the body. It's quick and it's non-specific. So that means it's not really looking for these specific peptide sequences that we'll talk about in a little bit. The adaptive immune system is really good at finding things that it's seen before and getting rid of them. The innate immune system is pretty much just on the lookout for anything that seems suspicious. So anything that comes in contact with cells of your innate immune system and structures of the innate immune system, it's just a general non-specific effort to stave off infection. So some of the structures of the innate immune system, the skin is the body's first line of defense against invasion by microorganisms. So sometimes we don't really think of the skin as an organ, or we don't really think of it as part of the immune system, but it actually is. It's a pretty important part because it separates the internal part of your body from the external. So it really is the first line of defense against, physical invaders into your body. And lysozyme is an enzyme that kills bacteria. It's found in tears, saliva, and blood. So a little bit in the blood, tears and saliva especially, just because whenever your body has openings, like openings in the skin, you want to make sure that we're protected. So that lysozyme is found in tears and saliva, and it helps kill the bacteria before they're able to actually get into those openings. And stomach acid is the same way. It may also kill pathogens before they enter the body. You have hydrochloric acid in your stomach. I believe it's pH around 2. So it's not very hospitable of an environment for any living organism. So a lot of times when we breathe in or when we eat, if there's bacteria on our food, if there are any other microorganisms on our food, the stomach acid in our stomach also plays a role in innate immunity, and it helps us kill those bacteria before we absorb them into our body. Neutrophils and macrophages are non-specific phagocytes. they engulf foreign microorganisms. So that's what a phagocyte is. You've probably learned in high school and college about phagocytosis. It's just the process by which one cell will take in something else. So neutrophils and macrophages actually eat these microorganisms and they digest them from within. And that's why they are called phagocytes. And there are a couple of different, there are a couple of other phagocytes as well. And then the complement system, there's not too much on the MCAT about it, but I did think it was worth mentioning. All my study materials mentioned it. But the complement system is just a group of proteins in your blood that non-specifically bind to foreign cells and promote an immune response. That's really all you need to know about the complement system. They're just the proteins that are floating around in your blood. If they come in contact with, say, a bacterium or something like that, they're just going to stick to the bacterium and they're going to alert your immune system that there's something else in there. So it's just another system that our body uses to identify foreign invaders. And if I haven't mentioned before, just please keep your mics muted. And then at the end of each section, you'll have a chance to unmute, or you can always type in the chat box. I'll do my best to answer questions in the chat box. Janelle is going to be the standby tutor. She's going to be a little bit late, but she'll be on in a few minutes to help us out as well. So continuing with the innate immunity, I put a picture to the right. It sort of outlines the lymphatic system. You can see all of those lymph nodes, those little bumps. And those are the lymph vessels throughout the body. We're going to talk a little bit about the lymphatic system at the end of the lecture. But for now, we're just going to continue on with innate immunity. So a major response of the innate immune system to fight infection is inflammation. Okay, so during inflammation, capillaries dilate and allow white blood cells and protein to leak out of the blood into the surrounding tissue. So that innate immunity, usually the first response, if you get a cut or if you get any sort of opening in your skin or any spot on your skin where there's a potential foreign invader, you're going to see this sort of inflammation reaction happening locally. And that really is just the body's non-specific way to try to kill whatever is being invaded. So it's going to, a lot of fluid is going to leak out. It's going to puff up. It's going to try to suffocate the bacterium. It's going to increase the heat in the area to try to burn the bacterium out. So that's a natural part of your body's innate immune system. And obviously, just like other systems of your body, sometimes it goes haywire. If you know anaphylactic shock, that's an inflammatory disease. So it pretty much just goes haywire and things that aren't supposed to be inflamed become inflamed. And then sometimes it's difficult to swallow, difficult to breathe, and it can result in death. So inflammation on a controlled level is a good thing. It helps us keep foreign invaders out and it helps us kill them before an infection happens. But like others, if it's going haywire, then it's definitely dangerous. So like I said, the capillaries dilate and it allows these white blood cells and proteins to leak out of the blood into the surrounding tissue. So we talked a little bit about this during the digestive system. We talked about it when we were talking about the renal system. It's just this theme in biology that whenever proteins or solutes leave a solution, the water is going to follow. So when we're talking about the renal system, if we're in the collecting duct, if we let sodium ions in, the water is going to follow. It's just how osmosis works. It's the same exact thing with inflammation. Capillaries are leaky by nature. They're usually pretty tight. And then when this inflammation occurs, there are a couple of proteins and a couple of different substances. We just generally refer to them as cytokines. And that's we're going to talk about it in a couple of minutes, but those are what the cells release and that causes the inflammation. So the capillaries actually sort of break apart and there are holes in the middle of them and they're large enough for these white blood cells. Really, the primary reason why they break apart is to let these white blood cells out of the blood and into the surrounding tissue. But white blood cells are so big that if we let them through, we also have to let these proteins through. So the proteins are going to also follow the white blood cells into the tissue, and just by osmosis, the water is also going to follow out of the capillaries into the tissue. And that's what causes inflammation. That's what causes this local swelling. And the swelling of tissue is known as edema. Fluids, proteins, and white blood cells in the surrounding tissue can be picked up and returned to circulation via the lymphatic system. So edema, our body usually handles edema by draining it via the lymphatic system. Sometimes when there is a malfunction of the lymphatic system, their edema can be pretty dangerous. I mean, I'm sure you've seen pictures of, people with feet that are just completely swollen from edema. And that's just because there's a dysfunction in the lymphatic system and it's not doing its job properly. Okay, so we talked a little bit about innate immunity, and now we're just going to go to the second part, which is adaptive immunity. So to the left, I put the structure of an antibody, which we're going to talk about. The adaptive immune system is the portion of the immune system that provides specific and long-lasting protection against foreign microorganisms. So the adaptive and innate immune system work together. It's sort of like two arms on the same person. One of them is a super quick response, so it gets to the infection as soon as possible and it just starts doing what it's supposed to do. And then the adaptive immunity plays the long game. It analyzes, it remembers, and it prepares for next time. So a major component of the adaptive immune system is the production and release of antibodies. And antibodies are also known as immunoglobulins. Each antibody has 2 light chains and 2 heavy chains. I think it's pretty important to know the structure of these antibodies. I do see them decently often on a bunch of review questions for the MCAT. So the heavy chains are obviously the longer ones, and the light chains are the outside ones on the 2. And this is just the general structure of each antibody. There are 5 classes that we'll go over, but most antibodies are like this. They all have 2 light chains, they all have 2 heavy chains, and on each of the chains, they have 2 separate regions. So if you look right in the middle, there's the variable region and the constant region. The variable region is also called the antigen binding site, and that's what varies specifically to recognize different antigens that come into the body. So this whole thing looks like a Y. These 2 tips of the Y are basically where the antibody is attaching to the antigen that it finds. This constant region remains constant for a specific antibody, but it changes depending on class. So there are 5 different classes. There, you would expect there to be 5 different types of constant regions. So there are only 5 constant regions, but the variable region will change for every single antibody that's out there because they have to attach to different antigens. So the chains are further divided, and we classify the antibodies into the 5 classes based on the sequence and structure of their constant region. So these are the 5 antibodies, the 5 classes of antibodies, and most blood antibodies are part of the IgG class. So IgG is actually the most common type of antibody. In your blood, it makes up about 80% of the antibodies that are in your blood plasma. And we could just take a look at these really quickly. So this first one over here, this sort of pentameric antibody is the IgM version. And the heavy chain, the letters at the end of them are just based on the heavy chain. So this is the IgM, it has a mu chain. This is the IgG, it has a gamma chain. That's not too important. Really the most important thing is just to understand that antibodies are separated into 5, and it would probably be worth your while to at least recognize that IgG is the main one that we deal with. But I would definitely try to commit these functions to memory as well. So IgM, the primary response fixes component, monomer serves as a B cell receptor. They are mostly B cell receptors, IgM antibodies. IgG antibodies are the ones that we're probably going to be talking about for the entire time. They're sort of like the prototypical antibody. They neutralize toxins, opsonization. IgA, those are antibodies that are secreted into mucus, tears, and saliva. So kind of before how we were talking about how lysozyme is in your tears and your saliva, it's the same thing. You have antibodies in there as well, because again, we're trying to cover our ground. Anytime we have an opening in our skin, we want to have as much protection as we can because it's an opening into our body. The IgE immunoglobulin is an antibody of allergy and antiparasitic activity. It's not super common, but it is implicated in allergy and antiparasitic activity. And then IgD is almost completely B cell receptors. So you'll find these IgD receptors, these immunoglobulins on the B cells, just to identify different pathogens. So the variable region of each antibody is different and constructed to fit a specific pathogen. Antibodies are produced by B cells. B cells are one of a type of cell that is part of the adaptive immune system. When an antigen binds to an immunoglobulin on an immature B cell, the cell differentiates into memory cells, which monitor the blood for future encounters with the same pathogen, and plasma cells, which are directly responsible for producing and secreting antibodies into the plasma. So immature B cell just means a B cell that's floating around your body that has not come in contact with its antigen, because every single B cell that your body creates from your bone marrow has a specific antigen that it will lock into. Some B cells will float around your body for a couple of years and will never come into contact with their specific antigen, and they'll just be broken down. But once a B cell comes into contact with its antigen for the first time, it's going to start dividing and it's going to split into two different distinct B cells. The first one, like we said, is a memory cell, and those are basically, it's basically just surveillance for your body. They're floating around your blood and they're making sure that we don't come into contact with that antigen again. If we do come into contact with the antigen again, then it attaches to it and it starts multiplying and it creates more plasma cells. Plasma cells are the ones that are directly responsible for producing and secreting antibodies into the plasma. So those are the ones that are actually releasing the immunoglobulins and the ones that are actually responsible for the immune response by the B cells. So those divide asymmetrically, they make two. And that's how we keep our immunity, basically, these memory cells. That's the theory behind vaccination. That's just how your adaptive immune system works. If we expose ourselves to an antigen, those memory cells are going to be floating around in our blood. So the next time they stick to an antigen that comes into our body, the immune response is super quick. The second time an adaptive response is triggered, it's extremely quick. The first time it could take days to weeks to mount full immunity from the adaptive immune system, but is quick the second time. Okay, so we're going to do our first review question. Before I do that, does anybody have any questions about what we just went over? You can use the chat box or you can unmute right now. Okay, if you think of anything, put it in the box. I'm going to launch the polls right now. If you don't see the poll pop up on your screen, you can click the poll button on the bar. These are anonymous, so please try your best. I'll give you a couple of minutes to do this, and then we're going to go over it as a group. All right, take another 5 seconds. I'm going to close the polls. Okay, so it looks like 100% of you said C, which is cool because that's the answer. Great job. So which of the following are part of the innate immune system? Pretty black and white. The IgG immunoglobulins they come from B cells, which are adaptive immunity. They're cells from the adaptive immune system. So one is incorrect. And from there, you can pretty much just cross out A, B, and D. Skin is also part of the innate immune system. We said it's the first barrier to protect against invading microorganisms. Macrophages also, those are the phagocytic cells of the innate immune system. They'll take in any sort of foreign invader, digest it. And stomach acid as well is part of the innate immune system. Excuse me, I just wanted to put these on here, like the skin and stomach acid one, just so that you really understand that the immune system is not just a bunch of microscopic cells. It's these larger structures, and they all contribute to immunity, specifically innate immunity. Okay, does anybody have any questions about that? Cool. All right, moving right along. We're going to talk a little bit more about the cells of the immune system. So I put the ones in here, again, that pop up often on the MCAT. There's a little guide right over here to the left. You can definitely take a read for those on your own time if you want some more information. You can definitely look it up, obviously. I always encourage you guys to use multiple sources when you're looking at the information. So we're just going to do a brief overview of all of these cells, but I do think they're pretty important. There's not a lot of content, like I said before in this lecture, but I do think the stuff that we talk about is pretty important. So it's definitely worth your while to commit them to memory. And these would be perfect for like flashcards. You can make flashcards. You could use the app Anki for flashcards. You can make, like paper flashcards. I think these are good things to do that with. So the first one we're going to talk about are macrophages. We spoke a little bit about them before. Those are the phagocytic cells of the innate immune system, and they originate from blood monocytes that enter the tissue to differentiate. So monocytes are another immune cell that actually have immune function, and they also have the ability to differentiate. So monocytes will turn into macrophages once they enter the tissue. And they were actually at one point thought to be the same cell, but we now know them to be two different ones. They can also release cytokines in response to a pathogen. And I mentioned cytokines a little bit before, and a cytokine is just a chemical signal released to improve the immune response in a specific area of the body. It's usually like small proteins or substances of that nature. There are a bunch of different ones, but you just really have to know that cytokines are chemicals that are released by immune cells, and they do improve the immune response in a specific area. So they'll recruit other cells over. Maybe they'll initiate an inflammatory response, but a lot of cells do release cytokines to signal other cells. And monocytes, like we just mentioned, they are also another phagocytic white blood cell, and they're the largest of the white blood cells. So monocytes have the potential to differentiate into macrophages and dendritic cells. Dendritic cells are another immune cell. These monocytes are created in the bone marrow. They circulate in the blood usually for 1 to 3 days, and then they enter surrounding tissue to differentiate. So they're doing a lot during their life. In the 1 to 3 days, they're sort of acting as a phagocyte. They're acting as a specific, like an active immune cell. And then in a couple of days, they enter the surrounding tissue and they usually become macrophages, but they will also become dendritic cells as well. That's a picture there to the left. There's you could see the nucleus is sort of split into different lobes. That's pretty common among immune cells, you'll see. So dendritic cells, like we just mentioned, there's a picture of a dendritic cell to the right. It's present in tissue that is in contact with the external environment. So dendritic cells are usually like in your skin, they're in your lungs. I think they are in your esophagus. So those sort of external tissues, the ones that are sort of lining the inside, but they're still considered external, that's where dendritic cells lie. So they also help with immunity right there. So they're derived from monocytes. And when they're activated by a pathogen, they head to the lymph nodes to aid the adaptive immune response. So they're sort of like the lookout. I don't think dendritic cells have too much activity, like sort of destroying pathogens themselves, but they're sort of the lookout. So they head to that lymph node, and the adaptive immune system, they'll usually like T cells or B cells there, that will, initiate an adaptive response. So that's why we really consider dendritic cells to be the bridge between innate and adaptive immunity. And dendritic cells can also release cytokines to communicate with other cells as well. So if one dendritic cell, for example, comes into contact with a pathogen, it can release cytokines to activate other dendritic cells around it. So it'll activate other immune cells around it, and it'll just sort of amplify that response. The signal will get back to the lymph nodes. The lymph nodes will probably produce T cells, and the T cells will take it from there. So it really is a concerted effort between the innate and adaptive immune system. So we have neutrophils. Those are also phagocytic white blood cells, but neutrophils are found in the blood. One of the first responders to damage caused by bacteria, environmental exposure, and cancer. So neutrophils are really fast acting. They're usually one of the first ones that are on site. They're usually responsible for, like, when you get a cut or you get something like that and you see it's healing if it releases pus. A lot of times when there's pus on a wound or something, someone will say that's a sign that it's healing, which is pretty true, because the pus is actually dead neutrophils. the pus is created by neutrophils that are pretty much doing their job and trying to basically decontaminate whatever wound there is. And they are the most abundant granulocyte. So a granulocyte is a white blood cell that has granules, and they degranulate is what the process is called, and it releases proteins that are inside of it. So a lot of the proteins have antimicrobial properties. Some of the granulocytes that we're going to talk about release histamine, which is the inflammatory agent that causes inflammation locally when it's released. But neutrophils, they're mostly phagocytic. And if you just keep in mind that they're pretty much the agent that causes pus and healing like pimples or healing bug bites or healing cuts, you should do well to remember neutrophils. Eosinophils are another granulocyte. They make up about 2 to 3% of all white blood cells. And this granulocyte releases multiple cytotoxic proteins. So that's its main method. They usually help combat parasites and some other infections in the body, and they do that by degranulation. So they degranulate, and the cytotoxic proteins will usually kill the parasite or whatever infectious agent is in the body. And they're also associated with allergies and asthma. Eosinophil levels, I know, like from a clinical perspective, whenever someone is being studied for anaphylaxis or any sort of allergy like that, they always check the eosinophil levels because it's an indication of like the patient's condition with allergies and asthma. I'm not too sure specifically about what the indications are, but it definitely is used just to gauge each specific patient's condition in regards to allergies. Okay, and 2 more of these granulocytes are basophils and mast cells, which are usually taught together because they're very similar. They are slightly different. Mast cells and basophils have extremely similar structure and function. Mast cells were once thought to be basophils that had migrated into the surrounding tissues, but are now known to be separate. So kind of similar to the macrophage monocyte thing. They are very similar. They have extremely similar functions, and that's really the main difference. There are other differences like the shapes, the amount of granules that they have, the size of their granules are a little bit different, but their structure and function is basically the same thing, except that mast cells are in the surrounding tissue and basophils are in the bloodstream. So both of them, when they degranulate, they release histamine during degranulation to improve the inflammatory response. So they're responsible for acute and chronic inflammatory diseases like anaphylaxis, asthma, and atopic dermatitis. Again, histamine, anything involving the inflammatory response is going to be involved in any sort of dysfunction like anaphylaxis. Asthma is also an inflammatory disease, and so is atopic dermatitis. So it would make sense that these are the cells that we would look at when we have issues, when we have these kinds of issues. Okay, then we have our natural killer cells over to the left. These are also known as NK cells, and they are cytotoxic lymphocytes. So they destroy intracellular pathogens and tumors. Intracellular meaning inside our cells. So up until now, we've pretty much been talking about like phagocytic cells and cells that degranulate that are basically taking care of these sort of intercellular pathogens. So like bacteria, parasites that don't really need to live in the cell. They could very well be in the bloodstream. They could very well be like in the extracellular matrix. Natural killer cells target intracellular pathogens. So like viruses, for example, they will actually kill the infected cell, like our infected body cell. So they target the infected cell or tumor and they induce lysis or apoptosis. They can induce lysis in a couple of ways. I know one of the ways they do is like they sort of like open aquaporins basically, and they allow water to go in and the cell basically bursts. Apoptosis, obviously we've studied, it has to do with, caspase cascades coming from the mitochondria. It's just cell programmed death. But natural killer cells do have a way to induce apoptosis into those cells. And they do not require an MHC1 proteins to target a cell. We'll talk a little bit about MHC. Those are basically, they just basically present antigen to the immune system. And a lot of cells need that MHC1 complex in order to identify what cells they need to destroy, but natural killer cells actually don't need to do that, which is why studies have recently shown adaptive activity in NK cells They're being implicated in cancer therapies. That's really where the main focus for natural killer cells and immunology is right now. There's a lot of buzz around natural killer cells and cancer therapy. But we do learn about them in the innate immune system, but they have shown adaptive activity. They formulate antigen-specific immunological memory to aid in fighting against reinfection of the same antigen. So it's not too well known, but they do show some sort of memory. So like if a cell was infected with the same pathogen, a lot of natural killer cells will be able to remember it and recognize it. Okay, so we're moving on to the adaptive immune system. We have the B cells. These cells we spoke a little bit about before. They create antibodies. That's basically what their job is. They are produced and matured in the bone marrow. So you can remember that we have B cells and T cells. B cells, B is for bone marrow because that's where they mature. T cells mature in the thymus. So B, bone, T, thymus. All of them are produced in the bone marrow, but the other ones the T cells are matured in the thymus, but B cells are produced and matured in the bone marrow. They present antigens for other immune cells and they release cytokines. So yeah, basically a lot of this function is shared between different cells. And a lot of these cells have multiple functions. So when immature B cells encounter an antigen that fits, they divide and differentiate into plasma cells and memory cells, like we spoke about before. Plasma cells produce and secrete antibodies into the blood. Memory cells have immunoglobulins with the same variable region, but they remain dormant until presented with an antigen a second time. The second response is quick and robust. B cells with the correct sequences are selected and multiplied in a process known as clonal selection. So clonal selection is definitely something you should be familiar with. Basically what it is think, for example, just say we have 1,000 B cells and each of them has a different sequence for a different antigen that they will recognize. If we have a pathogen that's coming in, and let me just define my terms before I continue. Pathogen is anything that is invading the body that will cause an infection or a disease. An antigen is specifically the protein sequence that is on the pathogen. So when I say antigen, I'm referring to specifically what sequence we're recognizing. When I say pathogen, I'm referring to the entire microorganism that is going to cause an infection. So clonal selection, say we have 1,000 different B cells and each of them will recognize a different antigen. When we have this pathogen enter the body, it's only going to react with one of those B cells. And then once that B cell finds the pathogen, it's going to start that one specific B cell is going to start proliferating like crazy and make a bunch of copies of that specific B cell. So that's what we refer to as clonal selection, because the antigen of the foreign invader is selecting the specific B cell, and that specific B cell is cloning so that it creates sort of an army to destroy any other antigens, any other copies of that antigen that may be in the body, or the pathogen that may be in the body. So I hope that makes sense. So moving on to the T cells, we have two. The first of the two we're going to talk about are helper T cells, and these are also known as CD4 cells or CD4 plus cells. They are also written as, and they're part of the adaptive immune system. They have a specific response. And helper T cells are super important. They are also sort of like a surveillance system. Their goal is to activate other cells of the immune system. So basically, whenever they're very, very good at finding foreign invaders. And when they find foreign invaders, they alert B cells and T cells. Because they're not super great at finding them, the T cells are really where it comes from. They release lymphokines and interleukins to aid in immune response. They're cytokines. Lymphokines and interleukins are signaling molecules used to recruit a local immune response. So again, same thing as cytokines. And helper T cells are the target of the HIV virus. So that's what HIV is. It enters your body and it starts destroying your helper T cells so that your body cannot recognize an infection when it takes root. So whenever we're looking at like white blood cell counts or T levels, when we're talking about HIV, we're talking about helper T cells. So obviously nowadays there are a lot of retroviral, antiretroviral therapies. We can keep HIV down to undetectable levels and even positive people. So people who are HIV positive can live very long, healthy lives and not transmit HIV to anyone else. But it's still it's still an epidemic in the world. And I think it might actually be considered a pandemic still. And it's definitely a hot area of research, especially like immunotherapy, specifically regarding the helper T cells. The helper T cells recognize MHC1 and MHC2 proteins. I believe they mostly recognize MHC2 proteins, but have just recently been found to also recognize MHC1. So MHC1, they're they're just proteins that sit on the outside of the cell. So MHC1 are present on all nucleated cells in your entire body. And what they do is they randomly take inner contents of the cell, they chop them up, and they display them on this MHC1 to the outside of the cell. So it's basically just sticking out the internal. So a helper T cell, for example, will come along, check it. If everything is good, if it's a part of our body, then it's just going to move on to the next cell. If it goes to one of these MHC proteins and it sees that there's something foreign, it can understand that this cell is infected, it's got to go. So it'll recruit another immune cell to destroy the cell. And MHC2 are specialized proteins that are found on macrophages and B cells, and they present chopped up antigens recently phagocytosed by the cell. So MHC1 is on all nucleated cells, and it just presents the inner cell's contents at random. MHC2 is specifically for phagocytic cells. They take it in, they chop it up, they digest it, and they knowingly show it to the outside, knowing that it's a virus, knowing that it's a pathogen. So the two are a little bit different. So MHC2 is not present on all nucleated cells. MHC1 is. And the other type of T cell are cytotoxic T cells. And they're also known as killer T cells, or CD8 or CD8 plus T cells. And they're just specific cells. They proliferate upon detecting viral proteins on a cell's MHC1 protein. So they'll go around and they'll basically just look at this MHC1 protein that we talked about, how it just chops up at random the inside of the cell and it presents it. So if a cytotoxic T cell sees it, or if it's recruited by a T helper cell, it's going to attach to the cell and it's going to induce lysis and apoptosis, very similar to how natural killer cells do. So it'll either cause the cell to burst, or it's just going to induce the cell to undergo apoptosis as well. Okay. Does anybody have any questions about that? It's not super in depth with any specific cell, but it is a lot. It is a lot of information. So I definitely would recommend either watching this video again. You can always go to the YouTube page, even watch it with the captions on, half speed, 3/4 speed. You can unmute if you have any questions. You can put it in the chat box. Okay. All right, so I'm going to launch the poll again. Take a minute. Which following cells are not phagocytic? Try to answer it without going back to your notes. And again, these polls are anonymous. So take a minute and we will get back to it. Nobody wants to take a guess? Cool. I'll give you guys another 10, 15 seconds to get your answers in. Okay, I'll open the poll. So 100% of you said B. So basophils are not phagocytic. Hold on one second. Yes. Okay, cool. Just wanted to double check. Okay, yes. So the correct answer is D. Just because all three of the other cells are actually phagocytic. Eosinophils are granulocytes, but they actually do have phagocytic properties. Macrophages are also phagocytic. That's their main function, and monocytes as well can differentiate into macrophages. But before they do that, they are also able to take in and digest other cells, other pathogens. But that's why we're here. We learn. So yes, the answer was D. Basophils are not phagocytic. They degranulate. They release histamine, and they recruit other immune cells to the location. And the histamine also causes a local inflammatory response. So that's their main function. Okay, and we'll do other structures of the immune system. So bone marrow, for example, we'll start with. Bone marrow, your bone marrow makes more than 220 billion blood cells in a single day, which I thought was really cool. Two types of bone marrow. There's red bone marrow and yellow, and they each produce different types of blood cell. So red bone marrow contains blood-forming stem cells, and yellow bone marrow contains stem cells that produce fat, cartilage, bone, all of that sort of connective tissue. So we do have stem cells in our bone. In our bone marrow, they divide, they become progenitor cells, and those progenitor cells will divide and further differentiate into the immune cells. Depending on what immune cells they become, they could further differentiate even more. So like if they become monocytes, then obviously they're going to become macrophages or dendritic cells at some point. So all blood cells, red and white, come from the progenitor cells, and the progenitor cells come from bone marrow stem cells. And B cells remain in the bone marrow to mature, but T cells head to the thymus. And these bone marrow stem cells are, again, another hot topic with like immunotherapy regarding cancer. They'll do like bone marrow transplants, if they can find sort of the focus of where the cancer is coming from, then they can actually try to transplant the bone marrow. And that is an area that they're looking into right now, which is cool. And B cells remain in the bone marrow to mature, but T cells, like we said before, head to the thymus to mature. And over here, we have some red blood cells. Over to the right, the platelets are also made. We're not really going to talk about them too much in this lecture, but those are just the clotting proteins that are also made in your bone marrow. And white blood cells, obviously, are also made in your bone marrow. Your appendix and your tonsils. I don't know why I put intestine there. I have to fix that. Both can be removed safely in humans without much consequences. I'm sure you all know someone who has had either one of them or even both of them removed. You can lead a perfectly healthy life without either of them, which led people to believe for a very long time that these were vestigial structures, when in fact they're not. Tonsils obviously have an immune responsibility, and the appendix, even more recently, was known to have an immune responsibility. So the tonsils are two masses of lymphatic tissue that sit at the back of the throat. They catch pathogens that enter the mouth and keep throat and lung infections away. So specifically when we inhale and when we breathe in, your tonsils basically act as a net. They can catch anything that's coming in, and they can notify lymph nodes and other spots in your immune system to mount a response. So if things are going into your mouth, the first piece of lymphatic tissue that they're going to hit, the first piece of immune tissue that they're going to hit are those tonsils. So they are decently important, but I mean, our body has a lot to its immune system. So when the tonsils are removed, we can still mount a pretty good immune response to pathogens that come in through our mouth, just because there are so many other avenues that it can go down. And the appendix was thought to have no function until recently. Like I said, even when I was in high school, we were taught that the appendix was a vestigial structure, that it was left over from thousands of years ago. But it does serve some functions. It's not super, super important, but it is made of lymphatic tissue, very similar to the tonsil. It does serve some sort of immune function. It basically just monitors the bacteria between your large and small intestine. So it's attached to the cecum, which is where your large and small intestines are connected. And it's basically just a breeding ground for bacteria. So it'll help the good bacteria grow. It'll help like your gut bacteria grow, but it can also detect when there are pathogens in there as well. So it does serve some small immune function, which is decent to mention. I don't think that'll pop up on the MCAT, but it's decent to mention. Okay, so we have the spleen, which is located in the left upper region of your abdomen. As you can see in this picture right over here, it's this little like 4 to 5 inch structure over there. It houses macrophages that help filter the blood and destroy old, unfit red blood cells. So it's kind of like a maze. Your blood goes through the spleen, and there are little macrophages all along the path. And if it detects any old, any misshapen, any sort of like blood cell that's not fit, it's going to break them down and it's going to recycle them. So it recycles the iron from the old blood cells and it sends it back to the bone marrow for hemoglobin production. Because any sort of mineral or any sort of metal that's in your body tries to do a good job at preserving it, because they're hard to come across, especially, in evolutionarily speaking, in the ancestral environment, they're pretty hard to come across. So your body has come up with some pretty cool tricks to conserve resources. And this is one of them. They recycle iron, your spleen, and it helps it create new red blood cells. So the spleen will also, instead of just filtering out old blood cells, it's also going to filter the blood for foreign microorganisms. So if they're detected, the spleen and local lymph nodes will increase lymphocyte production to combat the infection. So your lymph nodes work on a local system as well. Wherever your infection is in your body, you will be able to tell where it is based on which lymph nodes are swollen. So you may have noticed this before, you may not have, but for example, if you have like a cut or some sort of infection in the right side of your mouth, you're going to notice that the lymph nodes on the right side of your face are more swollen than the ones on the left side of your face, because proximity does affect lymph node function. And it's the same thing with the spleen. We have a lot of lymph nodes in our body. So the ones that are near the spleen, when they are detected, when microorganisms are detected, the lymph nodes are going to increase that T and B cell production, and they will combat the infection. And then we have the thymus, which is a pretty cool organ. It's located in the sternum between the lungs, and it puzzled scientists for a very long time, because they would always look at the thymus in adults. And by the time you're an adult, your thymus is not very prominent. By the time you're like completely old, it's basically just a heap of fatty tissue that does absolutely nothing. It's really only active from childhood to like your teenage years, because that's when your T cells are maturing. Once you reach adulthood, you basically have all of your T cells that are matured circulating your body. And the thymus plays a huge part in preventing autoimmunity, which are self-damaging immune cells, which we'll talk about in a little bit. T cell maturation occurs in the thymus during childhood until about the teenage years. So like I said before, a 75-year-old's thymus would most likely be nothing more than fatty tissue. And that was why it was so elusive to scientists that were trying to study it for so long, because they didn't really know that it was part of the immune system. They didn't know what the function was. So they were only looking at adults, so they weren't really thinking to look at children. T cells undergo positive and negative selection in the thymus. So the thymus is basically T cell boot camp. Positive selection means only immature T cells that can bind to MHC proteins will survive to maturity. So they have to be able to recognize those MHC proteins. If they don't, they're going to be useless, because that's what T cells do. They recognize these MHC proteins and they mount an immune response. So if they can't even recognize a foreign antigen, they're no good. So they would be destroyed in the thymus before they were released to circulation. They also undergo negative selection in in the thymus. So negative selection means only T cells that don't bind to self-antigens presented in the thymus will reach maturity. So the thymus is really cool in that it has these specialized cells in it that they display protein sequences that are found throughout your entire body. So you will find these different antigens in the thymus. You'll find like protein sequences that could be found in your eye, or protein sequences that will be found in your foot. And you can find them in the thymus because it's specifically where these T cells are made. So negative selection means if I put proteins that are found in the human eye, if I have a T cell in here that reacts with this and mounts an immune response, we're going to get rid of that immediately because that's an autoimmune response, and we don't want that. We don't want any cells that recognize ours, and we do want cells that recognize foreign. So T cells undergo positive and negative selection in the thymus, and it's super important that they do. Specifically, in my opinion, the negative selection, because autoimmunity is very dangerous. And I think that's a really cool process. And all of this is pretty much done by the time you reach your 20s. And then your thymus is pretty much inactive. Does anybody have any questions about that? I feel like they like to mention the thymus a lot on the MCAT I've seen. They never get too into detail, but they definitely do mention it a lot when immunity is mentioned. So definitely know those different organs that we just went over. Okay, so I'm going to give you guys a couple of minutes to answer this question. And we are really almost rounding the end of our lecture. So take a couple of minutes, think about the question. Let me, launch the polls. Hold on one second. Okay, the polls are live, so take a little bit. Remember, the answers are anonymous. If you don't have your poll clicker up, just go to that toolbar, click polls, and it'll pop up for you. They're anonymous, so just take a guess. All right, take another 10 seconds, get your answers in. Okay, I'm gonna end the polls. Cool. So half of you said A, half of you said I have no idea. And the answer is A. Cool, so let's take a look. A child undergoes emergency surgery and must have his thymus removed. Which of the following could be a potential outcome of this procedure? So B says the child will not experience any issues because the thymus is inactive, which is not true. We just spoke about how the thymus is active in children up until like their teens, maybe their early 20s. After that, the thymus becomes inactive, but a child undergoing surgery would still have an active thymus. The child will have reduced immunity because of interrupted B cell maturation. That's not true. B cells don't mature in the thymus. B cells mature in the bone marrow. The child will experience an increase in adaptive immunity. Probably not. Just because the child will have reduced immunity because of interrupted T cell maturation. That's really what is going to happen, because T cells are still under the process of maturing, and we're still selecting which specific T cells we want to let out into circulation. So if you remove a thymus while that's happening, we're not gonna have mature T cells, and the child would either be they would be immunocompromised, or they would have like some sort of autoimmune condition. But they definitely wouldn't experience an increase in like healthy working adaptive immunity. It would definitely be not good. So the child will have reduced immunity is the most likely outcome of that situation. Does everybody understand that? I can definitely explain that again if you need. It's just because we're removing the thymus before it has a chance to do what it needs to do. Okay, easy crowd tonight. Okay, so this is the last topic. We're just going to talk about autoimmunity and the lymphatic system very briefly. I put another diagram of the lymphatic system up to the right, and you can see how extensive it is. It's a pretty extensive network. You can see how it gets thicker and thinner at different parts of the body, and you see those little dots everywhere. Those are all of the lymph nodes. I think humans have like 500 to 600 lymph nodes throughout their entire body. So it's definitely just more than the ones that we can see visibly, on our neck and by our chest. So autoimmunity. A healthy functioning immune system will not target proteins in cells that are not foreign. So this is called tolerance. Our ability to not target our own cells is called tolerance. Often the negative selection that is supposed to occur for B cells and T cells does not, and self-reactive cells remain in the body. So that is for autoimmunity we're talking about. The negative selection is what we just talked about, where any cell that reacts with one of our own self cells gets eliminated. In autoimmunity, that does not happen, and B and T cells will react against like the host cells, against our own cells. B cells will undergo negative selection in the bone marrow, just like T cells in the thymus. Self-reactive cells will either undergo apoptosis or will be released as anergic cells. And anergic cells are just unresponsive. It's just another way that the immune system shuts off the cell rather than destroying it or inducing apoptosis. T cells will also undergo negative selection in the thymus and lymph nodes in a very similar process. So negative selection also does occur in the lymph nodes, not to the extent that it does in the thymus, but it does occur. Some autoimmune diseases that may appear on the MCAT are type 1 diabetes, rheumatoid arthritis, Graves' disease, myasthenia gravis, and celiac disease. So if you're interested in those, you can definitely look them up on your own time. Type 1 diabetes is usually the one I think that comes up the most. And then you'll see like other really rare diseases that'll come up. Like I've seen vitiligo on some of my studies, which is that autoimmune pigment disorder. But yeah, they're all pretty interesting, honestly. The lymphatic system, and that's really all I had to say about autoimmunity. There's not too much on it. It's basically, as long as you understand the concept, why it occurs, that's pretty much the most important part. I think if you have a general understanding of the immune system, then you should be able to understand what autoimmunity is and why it happens and why it's not good. So the lymphatic system, again, really quickly, is just the system in the body that circulates fluid. It is a one-way system, just like your circulatory system. And it acts as a suction pump to retrieve water and proteins and white blood cells from tissues. So like we were talking about before, when histamine is released, it causes those capillaries to leak. So we see white blood cells and proteins leak out into the cells, and then the water is going to follow them. The lymphatic system drains the cells and it dumps the fluid back into the circulatory system, and then acts as a suction pump. So we don't really it's not like a heart where we have the pump going. So it's basically just smooth muscle contractions and valves. It has a valve system so that there's no backflow. Smooth muscle contraction will cause the fluid to move forward, and then the pressure will just pull the fluid into the next chamber. And that's basically how the lymphatic system is circulated throughout the body. And the fluid that is in the lymphatic system is called lymph. intuitively. Lymph nodes are spread throughout the body and help filter lymph. So they're basically just dispersed throughout the lymphatic system. As you can see, even here to the left, these little dots that are through this orange, those are all lymph nodes, and this is the lymphatic system that's coming up the core. Small lymphatic vessels will merge into larger ducts, which merge into the thoracic duct, into the chest. Thoracic duct is the largest lymphatic duct in the system, and it empties into large veins into the neck. So the lymphatic system, this is the point where the lymphatic system meets the circulatory system, because basically that's the job of the lymphatic system. I mean, obviously it's going to collect these proteins. You're going to pass lymph nodes, so it does serve an immune purpose as well. The lymph nodes will be able to filter anything that's in the lymph, and if there is something that is invading, they can mount an immune response. But the lymphatic system is to get fluid out of the cells, and it's to retrieve water. It gets proteins back and white blood cells, it dumps them back into the circulatory system, and it meets the circulatory system in those large veins in the neck. So lymphatic vessels from the intestines also dump chylomicrons into the thoracic duct. So if you remember from the digestive system, chylomicrons are basically these little tiny lipoproteins that carry fatty acids through your blood, basically, because these fatty acids are not hydrophilic, they are hydrophobic, so they have to be packaged before they go in. So these chylomicrons get dumped from the intestines, and the lymphatic vessels can pick them up and dump them back into the circulatory system. That's how they get into the circulatory system. Okay, we just have one last question, and that's it for the content. So which of the following statements is false? Take a couple of minutes. Does anybody have any questions before we start this? You can unmute or type in the chat box. Sweet. Okay, give me 1 minute. I'm going to open the polls for you guys. Okay, the polls are open. All right, so take a minute or two, answer. Again, the polls are anonymous. We'll go over. All right, take another 10 or 15 seconds. Okay. All right, so 100% of you said the correct answer, which is B. So which of the following statements is false? We can go through it. A healthy immune system is one with tolerance. That is correct. Like we just discussed before, tolerance is our immune system ability to ignore are our natural cells. It's the ability to not induce autoimmunity, only target foreign cells. So that is true. That would be true. C is T cells that do not properly undergo negative selection in the thymus can go on to cause autoimmune damage, which is true because, again, negative selection protects against autoimmune damage. If we present in the thymus, if we present a T cell with proteins that are commonly found in your finger, and that T cell attaches to that, we're going to destroy it because of negative selection. We don't want an autoimmune response. So that's true. If they don't properly undergo negative selection, it's going to cause immune damage. T cells, okay. C and D. There are hundreds of lymph nodes throughout the lymphatic system. That is true. I just said that like a minute or two ago. I think there's like 500 to 600 total in like a full-grown adult in the lymphatic system. But B, the lymphatic system empties into an artery in the chest is not correct. The lymphatic system empties into a vein in the chest, which is correct, but it does not empty into arteries. It goes into veins first. And that's really the only reason why B is incorrect. And that's it. Does anybody have any questions about that question? Or questions about the rest of the lecture, anything we talked about tonight? That's it for the content. So if you guys have any questions, I will stand around for a couple of minutes. If not, have a great night. Thank you for coming.